Welcome everyone to week number 3. We've got a lot going on this week. You're going to learn about cells and how they work, and also you'll start your final project this week. So let's see what we've got going on. Okay, quick review of last week. You learned about pH, which is a measure of hydrogen or hydroxide ions, which gives you an idea of how acidic or alkaline an aqueous solution is. And that's very important because most cells like to have a nice neutral pH of 7, which is a balance of hydrogen and hydroxide ions. We learned about buffers, which are chemical systems that maintain pH. And this is very important in a lot of organisms because that pH of 7 needs to be maintained or the organism will suffer. This week we're going to learn about diffusion and osmosis. Diffusion is the principle where molecules move from areas of high concentration to low concentration until equilibrium is reached, or the molecules are just equally dispersed over the area. That makes sense. Osmosis is the movement of water across a semi-permeable membrane. Now, why is this important? Because every cell is surrounded by a membrane. Every cell that has ever been in history, every cell alive now, every cell in the future has movement of water across this membrane. And that is terribly important. Water is necessary for life, and that's because it needs to be in cells, especially to modify the heat production that's caused by all the chemical reactions that are taking place. You'll also learn about different types of cells and viruses. Well, what types of cells are there? First of all, there are bacterial cells, which are also called prokaryotic cells. The names are interchangeable. So bacterial cells are very small and they are fairly simple. They're surrounded by cell walls almost always, and they always also have a cell membrane. Their cell structures are very simple. They don't have little compartments in their cells like our cells do. They don't have a nucleus, they don't have mitochondria, etc., etc. So you're going to learn about bacterial cells. You'll also learn about eukaryotic cells. So that's like our cells that do have those little compartments. These cells are much bigger than bacterial cells, and they have compartments like a nucleus, like mitochondria, the endoplasmic reticulum. Oh, there are just so many more. Chloroplast, there's so many more. So those are eukaryotic cells, and they include the plants, the animals, fungi, and protists. And an example of a protist is an amoeba or green algae or paramecium, different organisms like that. Those are eukaryotic cells. And then we have viruses. Now, viruses are not considered to be living, but of course they're particles that infect us and plants and fungi and, yep, all the types of cells. So what are viruses then? If they're not living, if they don't have cells, they are little packages of genetic information. They either contain DNA or RNA. And a virus has to infect a host cell because it has no way to build any molecules. It can't make copies of itself. It doesn't consume food. It doesn't make energy. Viruses just don't do anything. They are packets of genetic information. So they infect host cells, take them over, and make the cell do what the virus wants it to do. You're going to learn about the structures in cells and viruses. There are membranes in some of these. Some viruses are surrounded by a membrane, including the coronavirus that is making its way around the world right now. All cells are surrounded by membranes. Some cells have cell walls. Some cells have nuclei. Some don't. So you're going to learn about those structures in cells this week and viruses. All right, let's talk about osmosis for just a minute. Osmosis, as I said, is the movement of water across a semi-permeable membrane. And in this example here, the sort of reddish thing there is supposed to be a cell. And you'll notice that outside of the cell, this solution contains 10% salt. Salt is considered a solute. A solute is something that's dissolved in the water, okay? So the salt is dissolved in the water, and it's 10% salt in this outside solution. And inside the cell is 20% salt. So the thing to realize then is how many water molecules, relatively speaking, are inside the cell versus outside. So if you only have a 10% salt solution outside the cell, then you have lots more water molecules outside the cell. You have less salt, more water. So what does that mean? The water is going to move into that cell and make it get bigger and bigger and bigger and maybe even explode. The solution outside of the cell is called hypotonic. There are fewer solute molecules out here than inside the cell. Hypertonic would mean that you have more solute molecules, and that means less water outside the cell. So which way is the water going to move then? The water is going to move out of the cell into the solution, and that means the cell will shrink or shrivel up. That is also not a good thing. Isotonic means you have the same number of solute molecules inside and outside the cell, and so water molecules move freely across the membrane, but there's no net gain or loss. All right, you will be simulating this with some dialysis tubing this week. I'm going to put a link to this little video, this YouTube video that shows you how to use the dialysis tubing. They do the experiment with molasses. We're using sugar water. It's basically the same thing. But this will give you an idea of how to use that dialysis tubing. I want to talk for a minute about COVID-19 because it's so much in the news right now. I'm just going to give you the basics of it. This is caused by a coronavirus. The virus's name is SARS-CoV-2. And what does that mean? Because you probably haven't heard that too much in the news. SARS is severe acute respiratory syndrome, and we all know that that's how this coronavirus affects us. CoV stands for coronavirus, and 2, we named it 2 because it's the second SARS virus that we have identified. All right, this virus causes COVID-19. Well, what does COVID stand for? CO stands for corona, VI stands for virus, and D stands for disease. So this is the disease process in a person. It's the coughing, it's the fever, it's the not being able to breathe, the tiredness, the loss of taste and smell, and all the different symptoms that you can have with the disease. This is the name of the disease. And SARS-CoV-2 is the name of this particular virus. Here's a picture of the virus here from the CDC website. The CDC website is a very good source for information. Well, what is this virus like? It is an enveloped virus, and that tells you that it picks up some of your membrane on the way out of your lung cells or wherever it is. So it's surrounded by a membrane, and this is actually a good thing because this means that alcohol-based hand sanitizers can destroy it because alcohol disrupts that membrane. You know how you can use alcohol to get fingerprints off of a, electronic device, or you can use it as a toner on your skin to remove oil. So you know that it does that, and membranes are made out of phospholipids, which are fats, and so hand sanitizers will work against this particular virus. This is an RNA virus as opposed to a DNA virus. It's non-segmented. That means it has one strand of RNA, and it is a positive strand, which you don't need to know what that means yet. And then there are lots of spikes on the outside of this virus, and they're used for attachment to the host cell. So we've got that going on right now. I've actually volunteered to be part of the vaccine trials, and I'm going tomorrow morning to see what that entails. So we'll see, and I'll let you know as I go through this. If you have any questions about the coronavirus or COVID-19, and you want to ask somebody, please feel free to email me, or we can chat, or whatever you want to do, and I'll be glad to help you out with that. All right, so the lab this week is going to take you time to do the potato part of the lab and the dialysis tubing. So there are two parts to this. You're going to chop up a potato and you're going to put it into some iodine, and then you're going to take it out and slice it open and see how far the iodine actually diffused into the potato segments that you have cut. The trick to this lab is that when iodine comes in contact with starch, which of course you know there's starch in potatoes, it forms a blue-black color. The iodine is normally a light brownish color, but when it comes in contact with starch, it turns blue-black. So you'll know if starch is present and how far the iodine has diffused by the black color. Iodine is corrosive. Be sure to wear your safety gear for this part of the lab. This is very important. You don't want to get the iodine on you. I know that you use iodine as an antiseptic, but it's not this strength. It's watered down with alcohol. So please wear your safety gear and be careful with this. There is 1 hour downtime during this experiment where you're waiting for this diffusion process to take place, so realize that. I'm giving you a hint on page 24 in the last column. It just says isotonic, hypotonic, hypertonic, but which, what? So if you'll write in there, in tube compared to the solution outside the tube, that will help you figure out what you need to write in. For the dialysis tubing, I'm going to link that video for you, but there's also downtime for that dialysis tubing for you to wait to see if the water rushes into the tubing or comes out of the tubing or just stays the same. So there's some downtime with that. I think that's also an hour where you have to wait. I think that's right. Before you check the volume again of the solution inside the tubing. So it is going to take you some time to get through these exercises. And then you have an exercise where you draw a bacterium using the instructions in the lab, showing the cell wall what it looks like. You expand it and show what the cell wall looks like. Is it gram-positive or gram-negative? That will be explained in the lab. You're going to draw a eukaryotic cell, and you're going to draw a virus. Now, you don't have to be a great artist. I certainly am not, but I can do this stuff. They show you what the structures look like that are involved. So this is not hard to do. And then what you're going to do is use the dichotomous keys that are also included in this lab to identify what is this bacterium that you've drawn that you just made up. What is this eukaryotic cell, and what is this type of virus? A dichotomous key is one where you have two choices. So it could say, in the virus identification, it could say, is this a DNA virus or an RNA? And if it's DNA, go to 2A. And if it's an RNA virus, you go to 2B. And then under whichever choice you have, then you're going to have two more choices. Choose 1 or 2. And then go down that path. So you'll see how this works. Sometimes in this key, there's 3 choices, which you don't have in dichotomous keys. But anyway, usually it's 2 choices. And so this is how you identify what you have. All right, the other thing that I want to mention is the experiment next week has to be started by Thursday in order for you to finish, because it will take you 2 days to germinate your peas in order to carry out the experiment. And it is extremely important that you don't use tap water for this. You need to use bottled water, distilled water, treated water, whatever, to get the chlorine out of it. Because if you put chlorinated water on top of your peas that are supposed to germinate, they won't. So be sure you have some of this kind of water. And even if you have tap water, you can use tap water. if you collect some in a big tub or something, big bowl, and leave it out for at least a day, and then the chlorine will evaporate from it. But do not use chlorinated tap water for this experiment. Okay, and then we have your final project. Milestone 1 is due. I told you last week to go to the Ask a Biologist website and figure out which type of experiment you want to do. You're going to have to come up with a research question. Okay, it's one of those if-then statements as a hypothesis. You're going to have to come up with that. And then you are going to tell me why you want to do this experiment. And here are 2 questions that you should answer. And answer them thoroughly. This is not one sentence. You know, these are paragraphs. Why is this idea, why is this concept, why is this question important in the grand scheme of things? Why is it important to know that water pockets form, brine pockets form in ice that's made of salt water? Why is that important? You're going to have to figure that out. Why is it important to know that you have fewer or more pain receptors on your fingertips than you do on your back? Why? Why do you need to know this? And what is known about this phenomenon? So you need to look up information about this and describe it. You need to be learning about this phenomenon, and you're going to describe what is already known about this. So that's your background information, and you need to do this research in order to do this milestone number 1. Be sure to follow the format using the guidelines and rubric for milestone 1. This is more of an outline for milestone 1, but you still need to include at least 3 sources of information and include that information in this outline. And if you have any questions, let me know. If you don't want to do one of these 4 experiments, remember that I told you the other 2, what were they? I had them. Breaking proteins was another really good one from Ask a Biologist website, and heavy water. That was also a good one. So you actually have 6 to choose from. And if you don't like those, then let me know, and maybe we can figure out something else. But remember, it has to be doable in our time frame. All right, I hope you have a wonderful week. And let me know if you have any problems or questions. I am here for you. Have a great week.